Hello. Good morning, everyone. Am I audible and clear? I hope I am audible and clear. Okay. I am Ami Susan Prasad, Faculty of Chemistry Department, St. Stephen's College, Uzhavoor. I am here to introduce our session conducted by our department as part of our college's International Multidisciplinary Online Conference, Quaesitio 22-23. And this program is intended to induce research culture among students. And our program will be inaugurated by a talk by a valuable resource person, Dr. Harindranath Kadavath, who is working as a senior scientist in USA. And I'm sure your knowledge will be enhanced by this session. And we, the Department of Chemistry, is not just happy to announce that not just research scholars and faculty is presenting, but also students are presenting in our paper presentation and participating in our session. And so to begin with, I invite our dear HOD, A.B. Jimson sir, to introduce our speaker, Dr. Harindranath Kadavath. Sir, please.
Good morning, everyone. I think I am audible. A warm good morning. Myself, A.B. Jimson, Assistant Professor and Head of Department of Chemistry. My duty is here to introduce our resource person, Dr. Harindranath Kadavath, who is an able man from the state of Kerala itself. He's a Malayali like us. And now he is working as a senior scientist in the Department of Structural Biology, St. Jude's Children's Research Hospital, Tennessee, US. By coming to his background, when I had a contact with him, I learned so many things that a resource person is such a talented man that he is a part of many research works and publications, including a lot variety of fields and including the nanoscience. He completed his graduation, post-graduation, and from the University of Connecticut, and later he went for a PhD at the Max Planck Institute, Germany. So, in fact, we are actually very happy to have such a resource person because Quaesitio is intended to improve the research culture and other methods in science for our science students. So, sir, here I welcome you to this gathering, and we are really happy that we have such an able resource person here. Thank you, sir. Please welcome.
Thank you. Is there anything else that you had to do, or should I start my presentation?
Excuse me, sir, can you please repeat?
I was wondering whether you have anything else to say, or should I start my presentation?
No, sir, you can start your presentation.
Okay, thank you. Yeah. Okay, let me share my screen.
Okay, sir. So can we start our presentation?
Oh, yeah. I was just figuring out the share my screen. Okay, I got it. Yeah.
Okay, sure.
Give me a few seconds. I think now there's a permission issue.
Okay, sir, sure. Really.
Okay, I'm sorry. I need to connect from another Mac. This one has a bit permission issues.
Okay, sir, no issue.
Just give me 1 minute and then I'll do.
Sure, sir.
Can you see the screen now?
Yes, sir, it's visible. We can see.
Okay, I'm sorry for the confusion. It's one of the computers I was using for the newest, and it has some permission issues. I'm sorry for that. So, okay. Good morning, everyone. First of all, thank you very much for the kind introduction, and thanks a lot for the invitation to present in your college. So I agreed when immediately, Bindu asked me like, sorry. Okay. So it's my pleasure to present in front of all of you part of my recent, it's over years, like I would like to give an overview of what I've been doing in the past, in order not to make it so difficult to follow for the graduate students as well as postgraduate students. Let's see. Yeah. So this talk I kind of, partially constructed in such a way that there will be like in slow. I would like to start with a kind of introduction. Of course, all of you studied NMR, and in terms of theory and some a little bit of the applications and so on. Of course, the bachelor's as well as master's level in our chemistry curriculum, you might be learning this in a little bit of organic chemistry point of view. But what I am presenting mainly in terms of the applications is, as you can see here, the biomolecular NMR. And usually we do it in vitro conditions that just like we do in an NMR tube or simple isolated conditions. But that's not the real picture that, that's in reality. So that's why people started doing in-cell NMR also recently, right, in terms of biomolecules. And so I'd like to give you an overview of this, okay? Before we start with, I recently moved to US. I joined here as as your HOD mentioned, like as a senior scientist. I joined here in St. Jude Children's Research Hospital in August. So until then, for almost 6 years, I was at ETH Zurich. So I'm still affiliated to ETH Zurich. So I present, most part of my research is mainly from Max Planck Institute, the one I'm presenting, and also mostly from ETH Zurich also, right? So all my slides are with that layout, and also I'm very much, personally attached to ETH Zurich. So nothing else with that, so that's why I prepare everything in this ETH template. So when you talk about, in terms of when you talk about the institutes in Europe, right, like especially the leading institutes, when it is the Max Planck Institute, I'm talking for you, the students, okay? The Max Planck Institute, or like there are several institutes in Germany, all over Europe. But when I moved to Zurich, to ETH has two campuses. It's the main campus is such as the center of Zurich, And this is at the uphill, like a few kilometers away from the city center, but this is in between the forest. This is a new campus. This is where I was working on. My office was located somewhere here. So these are both of them are really beautiful campus. As you see here now, the ETH center is at the city center, surrounded by the lake and the mountains. These are the Alps mountain that you can see here. I mean, of course, not my presentation is not about the geography of Switzerland, but I just wanted to highlight some important areas around ETH Zurich. So, and ETH Zurich is Eigennützige Technische Hochschule in German, but it's in English, there's a Swiss Federal Institute of Technology. And so far, there are 21 Nobel laureates from ETH Zurich. And, one of the first Nobel laureates was the Nobel Prize was in physics. That was for Einstein. I mean, all of you all know that he's well known for his mass energy relationship is equal to mc square, right? And so he received his 1921 Nobel Prize for physics. And over time, there are, as I said, like 21 Nobel laureates, and 2 of them were for the field of chemistry, and that too in NMR, right? So the 1991 Nobel Prize was Richard Ernst. He was working in the same department. I also had this opportunity to talk to him. Although he died last year, in the end of last year. However, I could interact with him, and he's such a nice person. This professor, and in 2002 for Jude Wittrich, both of them were he's in the chemistry department, he was in the molecular biology department. So the most important part is, so both of them were awarded this Nobel Prize for NMR research. So we have studied during your bachelor's onwards, you might have studied various spectroscopic methods and its applications and so on, right? So when you talk about now NMR, of course, it uses electromagnetic radiation, and this radio frequency range, in the radio frequency range, as you can see here now, this of long wavelength, that means low energy, which has a lot of advantages. On the other hand, there are more disadvantages, like there is less sensitivity and it demands more experimental time and so on. Nevertheless, when you go on again with this NMR field, right? NMR has advanced a lot. This was, this method was developed back in 1930s and 40s. So over time, so far there are 6 Nobel Prizes for NMR, and 2 of them are for, as I mentioned the previous slide, for Richard Ernst as well as Jude Wittrich, and both of them are from ETH Zurich. So, I'm just proud to say that, I could work in that environment and so on. Now, so the students, you can consider all these institutes for your future research and, other activities and so on. That is my point, maybe. So, I'm not going to spend too much time on the basic introduction of it. We know that, the properties of a spin, any unpaired electron, protons or neutrons, they possess a spin. It's usually spin half. So any atomic nuclei which are composed of protons, neutrons, and all, they all possess spin, right? So now the spin of a nucleus is determined by the number of protons and neutrons. And I'm sure that all of you are well aware of, the basic and necessary condition for a nucleus to be NMR active. There are conditions, right? So when you look at those nuclear spins, usually the nuclear spin quantum number can have values of 0, half, 1, 3 half, 5 half, and so on, right? So any nuclear spin that has a spin half, or some of them are NMR active, some of them are non-NMR active. For example, especially usually in biomolecules, right? In bio-NMR, we usually use 13 carbon, 15 and 1 nitrogen, 31 phosphorus, because our body is composed of amino acids and all other biomolecules, and most of the biomolecules are composed mainly of these 4 atoms, right? So of course you will have calcium, magnesium, and all these kind of metallic ions and so on bound to proteins, but these are the base mainly body amino acids, like basic structures composed of all these 4 isotopes. Even then, 12 carbon is not NMR active because of its nuclear spin, but others are NMR active. And as I mentioned again, it's not only spin half nuclei, these are NMR active, because these are more important in bio-NMR. But on the other hand, there are a lot of nucleus that has spin 1, for example, for deuterium spin 1, for 14 nitrogen that's also spin 1. There are many other nuclei like aluminum, vanadium, and so on, which has a spin of 3/2, 5/2, and so on, for example, right? I mean, your teacher, for example, Bindu, was working a lot on the quadrupolar nuclei back when she during her time at NCL Pune, for example. So there are also other nuclei, non-spin half nuclei, which are called quadrupolar nuclei, which is beyond the scope of this presentation. That's why I don't want to talk too much about that again. Right? So now, again, here, that usually a spin, the nuclear spins are randomly oriented in the presence of magnetic field. When you bring to the NMR spectrometer, in the presence of magnetic field, these are either aligned along with the magnetic field or across the magnetic field. Then comes the energy splitting, now the quantize, the Zeeman splitting, and so on, right? As I said again, like, I'm not going to spend much time here. I just want to say, okay, these are just want to remind you of the important aspects when you do the NMR, right? So a basic NMR spectrometer consists of, like, just like just in simple cartoonic representation, you can consider as a kind of as a radio station as well as a recording studio, right? So here we have a computer that kind of controls everything, all the electronics. So this is called NMR console. Usually there are a lot of amplifiers, signal generating units, receiver units, and so on, which is considered to be the heart of an NMR spectrometer. So this NMR console then is eventually connected to the NMR magnet. So which, of course, there is a probe head connected to this, and when you insert a sample here on top of the NMR spectrometer, so this goes and sits inside the probe head. It's called probe head, and which has an RF radio frequency coil. It can generate signals and receive signals and it receives back, and that's recorded here spectrum, right? So that's the basic things happening. And when you look at a simple experiment, the normal NMR spectrum is acquired in 2 steps. That's called preparation and detection. Preparation means you have to, apply a kind of do something with the nucleus. That's a basic thing one can talk about it. So you apply an RF radio frequency pulse followed by acquisition of a signal. So this acquired signal is usually time domain signal, which you cannot interpret. It's it's difficult to interpret this data. That's why you might have studied now again Fourier transform NMR pulse, Fourier transform NMR. Right, upon Fourier transformation, this time domain signal is converted to frequency domain signal, which you can analyze or interpret, and this very much analyzable data. Right? That's what's a very basic part of NMR's spectral acquisition. And now you might have also looked at the importance of chemical shift, the term chemical shifts, right? Because when you acquire this, if you look at this, the color code is for different spectrometers, 100 megahertz. Of course, when usually the magnetic field strength is specified in Tesla. However, these days, commonly we say the spectrometer is 100, 200, and so on, based on the proton Lama frequency. Okay, and now at this particular frequency, if you are trying to bring the positions of the signals in hertz, at all different spectrometers, they have different units, right? So in order to avoid that, the chemical shift term was introduced, and again, of course, I'm not going to details of this, and I'm sure you are well aware of it, right? And now all these particular frequencies, now irrespective of the magnetic field strength, they will have same chemical shift values, like as you can see here. This is one of the 1D spectrum of ethyl benzene, a simplest molecule, right? So after that, there is a lot of development in terms of NMR instrumentation, and one of the commercially available highest magnetic field strength of magnets is, this is the 1.2 gigahertz spectrometer of 1200 megahertz, and this is the second in the world that was available, that got delivered in Zurich in 2020, right, 2 years ago. Now, I had to acquire a lot of spectra on this, until I left Zurich. So this is actually, that is a kind of booster for the scope of various types of NMR experiments. Okay. And now, when we talk about multidimensional NMR, multidimensional experiments, so usually we deal with one-dimensional experiments, and you also might have seen the two-dimensional experiments, right? But sometimes these are not enough. So we need to acquire multi-dimensional, like 3-dimensional experiment, usually 3-dimensional, but there are also 4D, 5D, up to 7-dimensional experiments in biomolecules. So this is mainly because, the most of the time, it's not possible to correlate different chemical shifts by one-dimensional NMR. I'll show you in the upcoming slides what it means. So also it's difficult to analyze 1D NMR spectrum in the case of larger molecules because of the severe signal overlap. So two-dimensional NMR may basically provides information about connectivity and proximity, but even 2D NMR fails in certain cases. That case you have to go for 3D or 4D experiments, right? So on top of that, 3D structure determination of molecules, it can be a small organic molecule or large biomolecule or a DNA, RNA, and so on. If you want to determine the structure of these biomolecules or chemical compounds, one-dimensional experiment is not sufficient. So you really have to go for multi-dimensional experiments. Yeah. So to make it much more simple, I would like to show you this is an 8 amino acid long peptide. It's a region of the spectrum is shown here, right? Okay, even in this case, 8 residue long peptide, these signals are actually already overlapped. Now you have to look at the 1D spectrum, proton 1D spectrum of 76 amino acids long protein ubiquitin. So you see now the signals are largely overlapped. Right now you have to analyze. We forget about it to work with such a 1D spectrum. Okay, and that's the importance of bringing 2D or 3D NMR here, right? So when you look at the 2D NMR, of course, now there are the diagonal peak. If it is homonuclear experiment, there will be a lot of diagonal peaks as well as a lot of cross peaks, right? And these large peaks, as you can see here, this kind of contour plot is like these are the diagonal peaks, and there will be the cross peak corresponding to the protons, which are either spatially or through bond correlated. Okay, and now there are different types of 2D NMR. So the mechanism works either through bond interaction, magnetization transfer, or through space. So there are different types of experiments, and also there is one bond correlation experiments, as well as long range correlation experiment. As I said again, this homonuclear as well as heteronuclear experiments, and these heteronuclear experiments are widely common in biomolecular experiments, right? So I'll be showing some of those spectra in the upcoming slides. So you can have a look at those and see how do they look like, right? And now the most common 2D experiments, especially these are mainly used in organic chemistry experiments, right? Like COSY, HEDCORE, HMQC, HMBC, and so on. And OSI is an experiment that gives the spatial proximity of protons that gives you a structural information, right? With this, I also would like to highlight the very first two-dimensional NOSI that was acquired by Professor Anil Kumar. So he's actually, he was a retired professor at IIC Bangalore, but he did this, the first-hand experiments were performed by Professor Anil Kumar during his time at ETH Zurich back in 1980s, right? 79, 80, 81. This was the time he was there. And this led to the first Nobel Prize to Richard Ernst, right? And there are a lot of hundreds of peaks in this one and so on, right? This can provide structural information. And this very first 2D NOSI of a protein, and that that provided the further chance to explore the structural information of biomolecules. And that gave rise to the structure biology of NMR, the origin of NMR-based structure biology, right? So that's why they got the Nobel Prize for the first Nobel Prize in for the biomolecular experiments, right? And this, if you are interested in how this experiment was developed and so on, so this story is actually published by Professor Kumar in the Magnetic Resonance Chemistry with the title First NOSI. Okay, it's very interesting to read. Back, like you have to think about now, 45 years ago, how what were the circumstances, and that time NMR was not that developed, especially bio-NMR was not developed. So it was a kind of very important step in this field. Okay, so now with all these, if you have all the basic experimental and sample setup and everything, you can do a lot of things by NMR, right? So I'm going, of course, there are a lot of applications as you can see here, but when you talk about biomolecules and solution NMR, so the most important applications are in synthetic organic molecules and natural products. So, and then of course you can study biopolymers as well as any polymers, right? Like, so you'll be studying a lot of polymer chemistry and so on. NMR has a lot of applications in materials point of view as well as in polymer point of view. So you can study a lot of peptides, proteins, nucleic acids like RNA, DNA, a lot of inorganic complexes. During my master thesis, that was in NCL Pune back in 2007, for example. I mean, I led a lot of experiments in inorganic complexes and organometallic complexes, right? And so this has a lot of catalytic application. That's why it is widely used in catalysis. It's very important that, okay, you can also use NMR in drug development, right, to drug discovery, for fragment-based drug design and so on. Then material chemistry, lot of research is going on these days in the lithium-ion battery field. NMR has a lot of advances and contributions going on, and so on. Then you can also use in the food industry for the food quality control and so on. Right? So yeah, these are some basic applications of solution NMR. So when you come to the protein NMR, the biomolecular NMR, so it demands usually sample preparation is very important. Usually we need isotopically labeled proteins. Okay, so a normal amino acid, normal proteins are of, as I said, if there is nitrogen, carbon, these are not isotopically labeled. Of course, the natural abundance is there, but if you want to do experiment because of the low sensitivity of NMR method, you need to do isotopic enrichment. For that, you need to provide biomolecules in isotopically labeled form. So then followed by reference assignment, that means each peak corresponds to which amino acid, which atom that you have to do that reference assignment. Then we usually collect a lot of structural restraints and all other information with which you can perform the structure determination or any other experiment. Once you have the structure and the assignment, you can do a lot of advanced biomolecular NMR experiment that includes protein-protein interactions or protein ligand or protein drug interactions. You can study protein dynamics and so on. That brings a lot of variety of applications for biomolecular NMR. And now, just to, as a cartoon representation, I would like to, I just pulled out some of the important works that was published already in the past. So you can study protein dynamics. For example, if you look at the protein helical conformation of this one, this is in this orientation, right? This is helices. But when for the protein to be active, the upper form means there is no ligand bound. The presence of a ligand, the orientation, the conformation changes, right? So this switches back and forth with these two orientations. So that's called the dynamics of the protein. And also when RNA or DNA binds, there will be a lot of structural rearrangements and certain regions are moving, and that's called protein dynamics, the movement of different domains of protein, which is particularly important for protein functions, right? The physiological functions. So that how these proteins are moving and so on, or protein domains are moving, you can study. You cannot study with any other method such as X-ray crystallography. So NMR is solution NMR is one of the ideal methods to study all these dynamics, protein dynamics. And another case, for example, when a ligand binds to a protein, what happens to it and so on. So this we can study the excited states, which you otherwise cannot be studied by normal methods. We can also study membrane proteins. There are a lot of different types of proteins, cytoplasmic proteins, mitochondria, associated proteins, membrane protein. That means, a lot of cell proteins are associated with the membranes. For example, the calcium channel proteins, or this particular VDAC protein that's very important for, the cation calcium this is a calcium transporter. So and so on, right? So how to study the structure of these membrane proteins, interaction with other cytoplasmic proteins, and so on. You can again use NMR. So many other biophysical methods fails in this case, right? So NMR is very important in this particular kind of applications, right? And most importantly, if you want to study the protein misfolding diseases, so this includes Alzheimer's disease, Parkinson's disease, and so on. Many other biophysical methods such as X-ray crystallography, cryo-electron microscopy, or electron tomography, and there are a lot of methods, that provide structural information these days. So none of this can provide atomic resolution information other than NMR spectroscopy. Okay, so my PhD was largely based on the protein misfolding diseases, or especially focusing on tau protein and its relation to Alzheimer's disease. And part of that research I'm going to show in a few slides, in a few minutes. Okay, so protein misfolding diseases can be well studied by solution NMR. Okay? That's the take-home message of this one. So now when you talk about biomolecular NMR, there are everything is associated with the amino acids, like the proteins. So the basic building blocks, as you know, there are 20 standard amino acids. So everything is, what we are looking at is the atoms present in any of these 20 amino acids. And now if you look at the protein, as shown in one of the previous slides, the 1D spectrum of this 1D spectrum ubiquitin. And now there's a large issue of signal to noise that affects the sensitivity. And now there's a resolution issue, that means the line separation. Okay. Excuse me. So in this case, we need to do multidimensional experiments. And for that, mainly in biomolecular, we go for heteronuclear experiment. That means it can be heteronuclear single quantum coherence experiments. Usually we say that HSPC experiment. So we mainly look at the backbone NH or sidechain NH, Or we can also look at the 1H and 13C correlation. So backbone NH means you get information of the backbone N and H, when it forms amide bonds. And this is HSQC, an example of an HSQC spectrum of a folded protein. Folded protein means that, you might have looked at certain proteins like hemoglobin, that's one, or myoglobin. These are the classic examples of proteins which have, either alpha helix or beta strand conformation. So if any protein has an alpha helical or beta strand conformation, and their chemical shifts are widespread, as you see here, the peaks are well dispersed. But in the case of this is a structured protein. Okay, so now you are looking at the correlation of this amide nitrogen to this proton, as you see here. In the direct dimension of the proton, here is a nitrogen correlation. So nitrogen proton correlation. So each peak corresponds to one amino acid. Okay, so if you have 100 amino acids in your protein, or there will be 100 peaks present in this one, and each peak is specific for each amino acid. Okay, they have different chemical shifts, different chemical environment, and so on. Right? That's the and this kind of spectrum is known as a fingerprint of a protein. Right? If you have a similar number of amino acids and similar type of amino acid, but these are randomly arranged in different proteins. Okay. And now their spectra will never match. So that's why HSQC spectrum of a protein is known as a fingerprint of a protein. Now, whereas in the case of an unfolded protein, like that means there is no proper structure. It will look like something like this, randomly oriented in space in terms of structure. So there are chemical shift dispersion. For example, in this case, this is widely dispersed. And in the case of an unfolded protein, the chemical shift dispersion is very narrow. And again, we are looking at the amide and proton and nitrogen correlation in this case. And now the basic 3D structure determination of proteins, the steps involve sample preparation. That means isotopically labeled proteins. So there are different ways of producing this. Now I'm not going to, again, the biomolecular biology and biochemistry part of it. So if you get the protein, you can do an experiments of your choice. You can analyze the spectra. Eventually you can get the structure and many other things. You can do a lot of things, right? As I said, this is again a structured protein. The structure is solved by NMR spectroscopy. And now, once you have the protein in protein NMR, once you have the structure, or once you have the assignments of each peak, you can also do NMR titration. Titration means, of course, in chemistry, the titration you have to do a lot of titration during your lab practicals and so on, right? But in this case, what we do is like you take the protein in your NMR tube. So then if there's a ligand, or either you can look at do the titration with the protein titration or protein ligand titrations. So when you do this, there are those amino acids which are interacting with the binding partner, those their chemical shift will be changing because upon interaction with the ligand, the chemical environment of those amino acids which are interacting are changing. Okay, and now that leads to chemical shifts of this particular residue. On the other hand, those amino acids which are not interacting, they remain unchanged. So on the basis of this, you can determine the dissociation constant. You can study the kinetics of the protein interaction or protein ligand interaction. You can get all this information, KD, K on, K off, K exchange, everything. You can also get the thermodynamic properties of this interaction, right? Protein ligand interaction or protein binding. So the delta H, delta S, delta G, everything, which has a lot of relevance when you want to study the molecular mechanism involved in the protein or protein ligand interaction. All these parameters will help you a lot to extract a lot of information. Okay, these are some of the basic aspects, introduction I would like to give in terms of NMR, in particular to biomolecular NMR. So with all this basic knowledge, let's see how we can implement all this in a physiologically relevant system. Okay, so as I said, now initially I would like to go a few slides to show the molecular mechanism involving interaction of tau protein with microtubules. So the tau protein is very important in terms of Alzheimer's disease. Okay, so this is a neuronal cell. In the neurons, there are a lot of cytoskeletal proteins like microtubules, actin filaments, and so on. Now that in our body, different cells have the rigidity because of the cytoskeletal proteins. So one of the important cytoskeletal proteins is microtubules, and tau proteins are usually, these are small proteins, usually bind in microtubules and stabilize them, okay, as you can see here. So when something happens to the tau protein in terms of the mutations and as well as so other modifications, or phosphorylation, this tau protein detaches from the microtubules, and that leads to the formation of neurofibrillary tangles. So this leads to the formation of intracellular deposits. Okay, so this tau detaches from microtubules. It forms the deposits, which is considered to be one of the hallmarks in Alzheimer's disease, in addition to the extracellular amyloid plaques. So if you look at an Alzheimer brain, if you take a cells from Alzheimer brain, Alzheimer patients, the most of the time there are two types of deposit. This is within the neuronal cell. Inside the cells there will be neurofibrillary tangles of tau. Outside there will be amyloid plaques. These are also kind of cellular deposits. So the best method to prevent Alzheimer's disease to prevent all these happening. So ideally it should be like this. The tau protein should be always bound to microtubules. The amyloid beta protein should be physiologically active, and it should not form the plaques. That's the ideal condition. So it's in this case, especially in Europe, US, even in India, right? And these days, there are a lot of Alzheimer patients. So it is very important, for the research community to look at the molecular mechanism involved in the interaction of tau with microtubules. So if you understand the molecular mechanism, we can develop anti-Alzheimer drug or anti-Parkinson drug that can prevent these diseases, right? Protein misfolding diseases. That's the basic idea of this research. So I would like to show you a few slides how we performed all these over time. So now when you look at the microtubules and tau, so microtubules these are composed of internetwork tubulin proteins. These are large proteins actually, which has two subunits, alpha and beta subunits, and which forms a kind of cylindrical structure like hollow cylinders, and they function as structural components within cells. All our cells, irrespective of human or any organism. So these microtubules are the structural components within cells, as well as actin filaments and many other cytoskeletal proteins. Okay, so I'm talking about mainly microtubules here, and they are involved in a lot of cellular processes such as mitosis, transport of vesicles and organelles, and so on. Now, when you talk about human tau protein, which are the interacting partners of microtubules, there are 6 isoforms, as you see here. These are found mainly in the central nervous system. So these are mainly disordered. There is no proper structure for these proteins. That's why they are called intrinsically disordered. There is no structure at all. Their function is mainly to bind, nucleate, and stabilize these microtubules, which are important for the stability of microtubules. As you see here, as you can see here, some kind of small steps. The black thing. This is attached to this, and that gives the stability of these microtubules. Okay, and now there are a lot of things known in terms of the. If you look at the tubulin, there are a lot of anti-cancer drugs. The tubulin-bound statin and drugs. That means that certain things I highlighted here in this red or cyan, like red, purple, and blue color. These are some small molecules. Usually, these are used as anti-cancer drugs over time. People identify because this is involved in mitosis. There are a lot of cancerous properties. Tubulin is involved in cancer development and so on. So how you can prevent cancer? Sometimes you can provide these molecules. I'm not going to talk about that, but I'm saying so the structure, mechanism of action, everything is known for certain molecules, and also this protein that is shown in yellow color here. This also kind of their structure, binding mode, everything is known. At the same time, the structure and dynamics of the microtubule associated tau protein is not known. How they interact, how do they bind, which part of the protein is binding to microtubule, how do the stabilize microtubules, and so on. So the main reason is the challenges are there because of the large. This is so large, right? So it's large molecule by this tau microtubule complex, as well as I said, the tau is an intrinsically disordered protein. So this is highly dynamic. So the dynamic nature of the tau microtubule interaction leaves any other structural biology method like X-ray crystallography, chiroptera microscopy, and so on very difficult to determine the structure of this microtubule bound tau. So that leaves only NMR that can provide atomic resolution information. So to this end, we decided to do NMR spectroscopy, biomolecular NMR of tau in presence of microtubules and tubulin to understand the molecular mechanism involved in their interaction. Okay. So what I did in the beginning, as I said in the beginning, we use heteronuclear single quantum coherence experiment where you look at this is the amide bond. Okay, now this will be connected to another amino acid, and so on. That's how the protein is structured, right? So the basic primary structure of it. So now you are looking at the amide proton correlation, with the HSQC experiment, HN correlation. So when we take a protein, this is just a cartooning representation for you just to understand how this experiment works. So take each color as a single amino acid. Okay, and now when it interacts with the microtubules, so there, just those amino acids which are interacting with the microtubule, as you can see here, they experience chemical shift perturbation, chemical shift deviation, as well as loss in intensity. Okay. On the other hand, this violet color or purple colored and red colored amino acids are not interacting with microtubule; they remain unchanged. Those interacting with the microtubule, you see something. Okay. So that's in terms of the chemical shift as well as intensity loss. Now, when I do the this is the microtubules, the electron micrograph EM image of these microtubules. So now tau is bound to this, but tau is invisible because it's such a small protein. I mean, it's not so big protein, small protein, but compared to microtubules, tau is relatively small. So you cannot see tau here. So these are the microtubules, stabilized microtubules. Now I'm showing the overlay spectrum of tau with microtubules and without microtubules in blue without microtubules. So when upon addition of microtubules, as you can see here in the kind of the zoomed area, so there are some peaks disappeared or very weak, and they also see a little bit of slightly shifted. Okay, in terms of chemical shift. So now I analyze this variation of this chemical shift perturbation as well as intensities, mainly the intensities across the tau sequence. This protein has 441 amino acids. It's a large protein, and I identified 6 short signal motifs of tau as labeled as A, B, C, D up to F. And these short motifs bind to microtubules. We believe, and there are some more proof, but at this stage, we believe they bind at the common binding pocket in a dynamic manner. At a time, only one binds, but when this goes off, this binds and so on. So like that, it stabilizes the microtubules. And now it was also important to understand, here we know, okay, which of the regions, which region of the tau. So this has, for example, this projection domain or microtubule assembly domain. This region is not at all binding to this microtubule, as evidenced by the no intensity loss at all. But on the other hand, the regions with intensity loss are considered to be interacting. Okay, and now so that gives you only an idea of which of the aspects of this protein are involved in binding. Okay, usually this is a kind of stretch of 10 or 12 amino acid long short peptides are binding. Okay, that's what I am highlighting in this cartoon. And now you also want to establish a molecular mechanism involved in this interaction. Usually these segments are unstructured, completely unstructured. Okay. And now when it interacts with microtubules, there are some, I'm not going to the experimental details of this one. So I perform this kind of, this is very challenging system. That's why I would like to tell you. So this was, when we were doing this research, we were not sure that, okay, we can get a structure of it. However, I took it as a challenge because also my supervisor was, for example, that I'm not positive. Okay, yeah, he told, okay, just try it. So that gives. I'm talking now to the students, right? Just take all the challenges when you do research and so on. So this turned out like I could determine the structure of all these short sequence motifs within a divide and conquer strategy point of view. So I determined the structure of all these shorter fragments, and finally. I determined the structure of all these fragments with the method called transfer nosy, and that's even now when you look at it now, especially after 5-6 years after determining the structure of this tau protein bound to microtubules, this is so important. Okay, so this is there are physiologically not important information. Just a moment. Sorry, I was getting a call. I just had to disconnect it. Sorry. So with this method, I determined the structure of these all these shorter tau fragments, and we are approaching the molecular mechanism involved in the interaction of tau with microtubules. So it was always also important to know, okay, where does tau bind on microtubules? Okay, some people were suggesting in the past. If you look at the literature, tau binds at the outside of the microtubules or the outer surface, like this, something like this. Or some people are suggesting, okay, that tau binds the interior of the microtubule, inside, right? Especially at the Taxol binding site, the anti-cancer drug. This is an anti-cancer drug, Taxol. So where Taxol binds, that's the same place where tau binds, okay? Whatever, nevertheless, I just decided to do various types of experiments. So I used the already existing information such as the binding sites of Taxol, Bacatin. All these are the anti-cancer drugs, some of them, So for this binding site and structure is already known from the previous studies. So I use this information and perform some competition experiments. You remember 2 slides ago, I was showing a kind of intensity profile. upon interaction of tau with microtubules, you have this kind of black profile. And now when I add vinblastine or these molecules separately to, as you can see here, so tau is tubulin is taken here, so you have a black profile, and tau and tubulin with vinblastine, presence of vinblastine, I could see that, okay, there is like, the gain in intensity in this case. What does that mean? That means vinblastine, which binds here in red color. So if tau is binding here, so then when you add vinblastine to the tau tubulin complex, vinblastine is coming and displacing tau from this. So you have gain of intensity. On the other hand, when you add becatine, thalidomide, or colchicine, these kind of other molecules which are binding here, other regions, they have no effect. That means tau is bound here. The other molecules are binding at their binding pocket. So that's suggesting that vinblastine and tau share the same binding site. Okay. In order to further support this, I also performed STDX NMR experiment. So STD-NMR is one of the very important experiment that's called saturation transfer difference. So this is mainly used for to see whether a molecule is binding to a protein or not. So you take a reference spectrum, that means reference spectrum of only the ligand, like proton A and proton B. You have two signals, and now when it binds, you will get signal only for the proton which is in direct proximity to the protein. You see, in HB there is very less signal. So that means this ligand is binding. Whereas it's a non-ligand, like a non-binding ligand, in the absence of this protein, of course, there is a signal, as you can see here in the case of a reference. But in the STD-NMR spectrum, there is no signal because that means there is no binding. This works with, there you apply magnetization to the protein, and that magnetization is slowly transferred to the proton, which is very close to the protein, which is possible only if it is binding to this. So this is, when you look at the pharmaceutical industry, if people do NMR spectroscopy, especially to develop drugs, a lot of drugs that is widely available these days. One of the methods people are using in pharmaceutical industries is NMR spectroscopy. If they do NMR spectroscopy experiments, the most important experiments is always STD-NMR. It's a very simple experiment that was discovered back in like late 1990s, but this turned out like very important and most powerful tool for NMR-based drug discovery. Of course, I'm not doing drug discovery here, but this method was in a similar fashion applied to all these kind of small molecules I listed in the previous slide. So I performed this experiment with tau fragments and all these anti-tubulin, anti-cancer drugs, vinblastine, colchicine, becatine, and so on. So this suggests that when I performed the competition experiment based on STD-NMR, so I could see that again vinblastine is the only molecule that competes with all these tau fragments, suggesting that vinblastine and tau share the same binding site. Okay, and now to further support this, when you do NMR-based structure biology or any structure biology experiments, most of the time we need to provide additional information, right? So we also wanted to do other than NMR some other methods, so that's why we went doing mass spectrometry. So basically what we did is a chemical cross-linking of tau with tubulin, chemical cross-linking. So the primary amine, so lysines, amino acids such as lysine, which have primary amines, like NH2 is a side chain. So this BS3, this is the small molecule here, right? So the BS3 is the abbreviation of this one. So BS3 when cross-linked to this amino acid, like only it binds only to, it will cross-link only lysine amino acids, okay? So you get something here, and when we do the in-gel digestion and mass spectrometric analysis, we really understood that, okay, if you look at the tau from 1 to 141, there are several lysine amino acids. However, the cross-links that we identified from the mass spectrometric analysis involved only lysine amino acids in 225, 243, and so on, which as highlighted here, which falls within the binding hotspots I identified in the previous interaction studies, suggesting that, and also the cross-link on tubulin was found only for lysine 338 as well as lysine 336, which is very near to the vinblastine binding pocket. So all these mass spectrometric analysis further supported our NMR-based investigation, identification that, okay, tau binds at or near the vinblastine binding site. Okay, with this we proposed a binding model of tau on microtubules that, okay, tau binds the interdimer interface. This is one dimer, alpha beta dimer. This is another alpha beta dimer. So tau binds the interdimer interface near the vinblastine binding pocket. Okay, suggesting that it binds and promotes a straight protofilament conformation. Vinblastine, on the other hand, promotes a different. They bind at the same point binding pocket. However, it has a different function and different mechanism of binding. Okay, so this is actually a very important study in terms of the tau microtubule research, especially in the Alzheimer research field. And people are still doing a lot of research in this field, and this is only part of our contribution. No? So now, not only microtubules, there are also actin filaments, which are also consists of cytoskeletal proteins. So when tau binds to actin, on the other hand, it gets structured again. But in this case, for example, it gets alpha helical conformation. When tau binds to microtubules, on the other hand, it gets a kind of well-defined turn-like conformation, right? So same protein tau binds to actin as well as microtubules with two different conformations. So this is a multifunctional approach, but different mechanism involved in the interaction. Now, we can also do a lot of other things. So far we have looked at the molecular mechanism involved in the tau actin or tau microtubule interaction, which is very important in especially Alzheimer's disease. Okay, but this is only to understand the mechanism. But as a collaboration, I was also involved in a lot of other research activities. For example, there's a small molecule, as you can see here, we call it like BSC 4090. So methoxy derivative, that's most important functional group of it. So with this, I'm not going to do the details of it. What I would like to tell you is this small molecule that you can use as a kind of biomarker to detect early detection of Alzheimer's disease. Okay. So what happens is you can take a kind of a little bit of sample from your nose. And if you are an Alzheimer patient, like even in the early stage of the Alzheimer disease, and if you bring that sample that's taken from your nose and in contact with the small molecule, that gives a kind of fluorescence, because of their binding interactions and so on. And that we again supported this development of this biomarker in terms of NMR spectroscopy. Again, I performed the saturation transfer difference NMR experiments and many other methods here, but the only highlight is you can do a lot of things by NMR. That's the take-home message. And now, just to conclude this part, okay, I didn't finish yet. I need another 10 minutes or so. So this is just to conclude this first part of this one. We identified the small, short tau motifs that interacts with microtubules. The tau binds to microtubules in a specific turn-like conformation, and this binds at the interdimer interface of alpha-beta tubulin heterodimers. And also tau regulates microtubules and affecting crosstalk, but with different mechanism of binding. Okay, and now these are all done separately. We took the proteins as well as the protein binding partners, small molecules that is interacting with them in NMR tube separately. Okay, now we established a mechanism. Okay, we propose there's a mechanism involved in their interaction. This can be widely applied for the Alzheimer research or drug design, anti-Alzheimer drug development, and so on. So that's what we propose, but of course on the basis of the in vitro conditioning experiments. Okay, but in reality, it's also important to look at the experiments in cellular environment. Why this is important? In vitro conditions, as you can see here, when you take the sample in an NMR tube, taken in a buffer, the trust protein is very much isolated. However, in reality, in cells, things are much different, right? If you look at the interior of a cell, the intracellular environment of eukaryotes, mostly eukaryotes, right, of mammalian cells or any eukaryotic cells, these are of complex heterogeneous nature, containing a lot of cellular machines. That means ribosomes, mitochondria, a lot of protein filaments are there, like microtubules, actin, the one I was mentioning in the previous slides. All these membranes, there are a lot of membranes, and you have the nucleus. So when you take the protein, in reality, this protein is present in all these heterogeneous conditions. Okay, so this is also important. It's also very important to understand the molecular mechanism of protein interaction or protein ligand interaction in a cellular environment. And one of the ideal methods to study this is in-cell NMR. Okay. And now, if you look at the in-cell NMR, the first in-cell NMR spectroscopy in general started like more than 5-6 decades ago, right? 7 decades ago. In biomolecules, people started working on, let's say, 40 years ago. but all the advanced NMR experiments in terms of biomolecules, they started doing it in 1980s and 1990s. Okay, so but
Excuse
when
me?
people started
Excuse
realizing
me?
that okay, you have to do everything in a cellular environment. That was back in 2000. Okay, people started doing in-cell NMR research, and that was mainly started doing it in bacterial cells.
Excuse me? Sir, your
Yes.
screen is not shared.
Sorry.
Your screen is not visible.
So it was not visible from the beginning, or
No, just now.
okay.
Can you share it again? Yeah, okay. Mic is not on.
Okay, it's clear now, right?
Yeah, right.
Okay. Again, it's visible, right?
Yes, visible.
Okay. So, if you're, I hope you're not lost, in between the presentation. So I have only a few more slides then, about in-cell NMR, then I think it will be more fine with that, like I'm done. So I just want to give you an overview of what's also, have you seen the previous slide? Did you see this slide when I was talking about?
Yes.
Okay, so just want to highlight you that okay, it's so important to do in-cell NMR because in vitro conditions, proteins are taken in isolated conditions, but in a heterogeneous environment, proteins are present in presence of lot of cellular organelles and other protein binding partners present in cytoplasm. Okay, and now an ideal method to do this is in-cell NMR, and in-cell NMR was first reported in 2001 in that too only in bacterial cells. Since then, a lot of developments are there in cell NMR field. That's mainly in the developments are mainly to do everything in eukaryotic cell because bacterial cells is the minimalistic point of view. But there are, if you want to study a lot of protein functions and mechanisms in humans, especially for drug development and so on, you have to do it in eukaryotic cells, right? So then people started working on yeast, xenopus insect cells, followed by, after some time, a lot of people started working on mammalian cells. It's a kind of cartoon representation of mammalian cells. So what we had to do is, we had to bring the isotopically labeled proteins to these mammalian cells. There are a lot of methods. So one of the common methods that I use, or I've been using or in our group back in Zurich, mainly we use electroporation. That's one of the simplest and less harmful method for cells. Cells won't be, because with several other methods, not all these are some of the selected methods, so cells might die or part of the cells might die. But electroporation conditions, even after electroporation for 5 seconds or so, cells are still alive. There is no problem. With this method, we bring the isotopically labeled proteins to cells, and however. So far, right, if you look at the recent past in literature of in-cell NMR, only a few proteins were studied in mammalian cells. That too mainly was studied in the case of protein alpha-synuclein. Alpha-synuclein is again like kind of unfolded protein. That is a culprit, like that's mainly involved in, it plays a huge role in Parkinson's disease. Okay, when tau protein plays a significant role in Alzheimer's disease, alpha-synuclein plays a significant role in Parkinson's disease. These two are intrinsic disorder proteins that's widely studied worldwide by several research groups. So even by in-cell NMR, people started looking mainly at alpha-synucleins, right? So much more known about for several folded proteins and normal proteins. people were not so successful because of the experimental limitations and lack of technology and so on. That's the time we started doing again in cell NMR, back in 2017, like 5-6 years ago. So our idea was to do, you produce labile proteins, you bring to the electroporation, by means of electroporation, we bring to the mammalian cells, because we wanted to look everything in mammalian cells, and take these cells, mammalian cell electroporated, protein electroporated cells. Now you see, these proteins are inside cells, you pack into NMR tube, you record the spectrum of your choice, depends on what you would like to study, and look at it, right? And then based on that, based on the type of spectra that you accurate, we can study the structure, ligand binding, or protein interactions, protein dynamics, everything in cells. Okay. We studied, we looked at several proteins. Okay. So now I am showing here the spectra acquired in the case of 5 different proteins. So prothymosin alpha, I abbreviate as PTMA. This is also involved in cancer. Alpha-synuclein is involved in Parkinson disease. GB1, ubiquitin, and PDC domain, and so on. These are different folded proteins. So the spectra shown here in blue are acquired in in-vitro conditions. That means you take the protein in buffer in NMR tube. Nothing else is present there. But this we bring to mammalian cells. Now you record the spectra. Overall, they look the same, at least the same pattern. So that means the protein fold, everything is the same. However, we decided to look at everything in detail. Okay. Now, in the case of initially, I wanted to look at the protein GB1. So, in vitro condition, you have always only for leucine 7, threonine 18, tyrosine 47. These are all amino acids present in the particular protein. Okay. So, in this case, we started looking at the peak pattern of this one. So, ideally, we are expecting only one peak for one amino acid in the fingerprint region. But whereas in cell spectra, you see, this one particular peak is split into now 3 peaks. Okay. In this particular cell, maybe 4 peaks, and so on. So I mapped this onto the 3D structure of GB1. So whatever changes are happening, more changes like threonine 20, there are 4 peaks, which is present somewhere here. The threonine 20 is here. So all these amino acid residues marked on red or orange are showing more of this kind of, I call this as this kind of, multiplication of peaks, multiplicity of the resonances. And now this kind of multiplication of peaks that can be due to motion of residues at or motion of these amino acids at millisecond time scale. That's called the protein dynamics. Maybe it can be due to the chemical modification of these residues. Residues means amino acid residues. Okay, individual amino acids are called residues, and they might feel different intracellular environment. That means maybe part of this protein is present near the nucleus, part of this protein is located near the mitochondria, some of them are lying around within the cytoplasm, here and there. Some of them are binding to different binding partners or different proteins and small molecules and so on. There are various potential possibilities. We don't know. We have no direct information on this. We got it, but I'm not going to show all this information. That will be too much to discuss today. So again, I also looked at the intensity variations of this. Again, some of the peaks are amino acids are showing reduction in their intensities in the spectrum. Some of them are not. Again, I mapped all these residues which are showing the reduction in intensities by taking the intensity ratio of the red divided by the intensity of the blue. That's what is plotted here. Okay. So again, I mapped on this. Similar amino acids are involved in all these, and this loss of intensity of these amino acids or loss of intensity of these peaks corresponding to individual amino acids, that can be attributed to motion of these residues at now microsecond time scale. So if you talk about NMR protein dynamics by NMR spectroscopy, we talk about different time scales, so millisecond time scale, microsecond time scale, and so on. So how fast or how slow the amino acids or the atoms are moving. We can basically, the take-home message is we can look at all this information by biomolecular solution state NMR, right? You can also do solid state NMR of biomolecules, but that provides a different information. So, and also this also suggests the transient interaction with intracellular binding partner. That means this protein GB1, when present in cells, they are able to find their physiological binding partners. It can be binding partner A, B, or C, we don't know. But there are several proteins, they are interacting with these molecules, and that's how our physiological functions are happening, right, within cells. All these are possible, okay? And now I also looked at, because of the time limitation, I'm not going to spend too much time on this. So, however, I wanted to know whether these interactions are because of unspecific interactions because of the crowding agent or not, or whether there's an influence of the pH. So I performed the pH titrations from pH 5.5 to 7.5. Why I'm talking about it is in endosomes, exosomes, mitochondria, different regions, sometimes the pH can be slightly different, right? Because of different compartmentalization. So is there any influence of that? Or different compartments might feel different salt concentration. Usually our, in physiological salt concentration is around 150 millimolar, altogether potassium chloride and sodium chloride. If you take a normal cells, it can be a little bit more or less at different regions of the cells. So that's why I performed the salt titration from 0 millimolar to 150 millimolar, the maximum salt concentration available in a cell. But there is no response of these chemical shifts with respect to this. If there is a change in response with respect to the pH or the salt titration, a change in salt concentration, you expect a kind of linear relation with respect to the chemical shift observed in this case. Okay. So the take-home message again here is, okay, there is no influence of pH, salt concentration, or crowding agents in the observation that we have found in the case of the insulin NMR spectra of various proteins. Okay, this was performed for all other proteins, all these experiments. And now I also would like to show, I'm finishing in like 5 minutes. Okay, I'm sorry if it is again too much. So the PDC domain is another important protein. It's involved in part of a domain, the domain of a part of a protein. So PDC domain, that's a kind of general term for a particular type of protein domains. Again, you have to think about a alpha helical and beta strand conformation. I'll show you that. So this is a kind of PDC domain of human tyrosine phosphatase, a particular protein. It's a very important protein. So when I look at the insulin NMR spectrum of this one, in vitro, you see again for each amino acid, like histidine 71, lysine 72, alanine 74, threonine 70, isoleucine 20, glycine 90. For example, selected 6 are shown here. For all these, there's only one peak. Okay, this peak and this blue peak correspond to another amino acid. So in vitro condition, there's only one peak corresponding to each amino acid. Now, when you look at the insulin NMR spectrum in red, there are a lot of additional peaks present, right? So when I map this multiplicity again onto the 3D structure, now these changes are located on the beta strand, this particular beta strand, starting from glycine 19, isoleucine 20, and so on, and also at the alpha helix, histidines, alpha helix comprising histidine 71, valine 75, and so on. Okay. Now, what is the importance of this particular region? So I also looked at the intensity variation. In this case, again, I mapped on this 3D structure. This intensity changes as well as multiplicity, as you could see in the multiplicity also. These are again located around this area, as you see here, the beta strand as well as the alpha helix too. Okay, what's the importance of this? So when you look at the structure of this PDC domain, so there's a gray color, I highlight here a kind of short peptide. So this particular structure is a complex structure of, there's a C-terminal peptide coming from the guanine nucleotide exchange factor. This is very important protein again, so PDC domain is one protein. The one I shown here in kind of orange, green, and red. And the one is in gray is only an edge of the C-terminal tail of another protein. So this is a complex, okay? So this particular region in gray is coming and binding here. So now I titrated this. What I did is like I performed a NMR titration of a peptide VSAV, only take this peptide separately in vitro condition, okay? Now I'm taking this protein alone in an NMR tube. Now I add this particular peptide, that's the NMR titration, okay? I add systematically. In blue, only protein is present. In yellow, I add the protein and ligand in 1:1 ratio. In magenta, I do 1:2 ratio. In green, I do 1:3 ratio. And upon successive addition of, in stepwise addition of the ligand, I see chemical shift perturbation, suggesting that this particular ligand is interacting with glycine 19, isoleucine 20, and threonine 70. Okay, threonine 70 is here, present here. So that means this ligand is binding. Now, if you look at these changes here, this is the third panel here that I'm showing here. This is in vitro condition, but the chemical shift perturbation, the chemical shift, the shift of the peak from blue to red is in the same direction as you see in vitro spectrum, okay, in cells also, suggesting that even in the presence of, when we do insulin NMR experiment, when we do bring PDC domain to cells, we provide only the PDC domain into living cell, mammalian cells. We are able to, this protein is able to see its physiological binding partner present in the cell. Okay, we don't provide this binding partner at all. We provide only PDC domain. So when we provide, so see, this is applicable for any other protein, right? Even the one I showed in the previous slides also, or there are a lot of proteins which I don't discuss here today. So we see this kind of information, like, this is kind of direct proof that when we bring PDC domain to mammalian cells by means of electroporation, this protein is able to find its physiological binding partners. Okay. We also went on doing many other things. One of the important applications of solution and a biomolecular solution NMR is to determine protein structures also, right? So when we do in-cell NMR, so far many group they were not successful in determining doing in-cell NMR mammalian proteins in mammalian cells. So on top of that, people, very less number of groups were able to show structural determination of proteins in cells, living cells, right? So we wanted to further explore that. We were able to, I just want to give you a kind of, highlight of, we were able to do the protein structure determination in mammalian cells. That was a kind of very, we were very ambitious, and it was very successful experimental method that we implemented in the lab. So another important protein was prothymosin alpha, the one I showed in the beginning. So this again, it's an inducing disorder protein. This is involved in cancer. I'm finishing in 2 minutes or 1 minute. So involved in cancer. So if you look at the protein in vitro, overall protein present in vitro or in a crowded environment, these three are in different mammalian cells. HEC means human embryonic kidney, COS7 cells or 18-780 cells, neuronal cells, non-neuronal cells. We tried many cells. Okay. So overall, there's no difference. Now, when you look at these regions, for example, this is in vitro, when the protein is taken only in buffer, in isolated condition. But when you look at different cellular conditions, in this case, these two peaks are strong, these two are weak. But in this case, this is the other way around. These two are similar. And so on. This is like that. This is still like a kind of ongoing research part. I just want to show you, we can do many things. So this kind of small differences, when we analyze it in detail, residue by residue, amino acid by amino acid, we can understand a lot of things that's going on, and we could explain a lot of things. So which is beyond the scope of this presentation now, right? I mentioned this prothymosin alpha, which is a lot indirectly involved in cancer progression, right? So if you can establish a mechanism involved in their interaction with the histones, with binding partners, and so on, this will be part of the anti-cancer drug development. And I was also involved in a lot of anti-cancer drug development projects, which again I am not showing here today. That's very important, like protein KRAS and so on. So I just would like to conclude here. I think I understand that it will be kind of too late for you now. I heard that you have a kind of tight schedule today. There are a lot of presentations. There are a lot of people to present and so on. Okay, so within a minute, I think I'll be able to conclude everything. So again, the second part, when I talked about the in-cell NMR, so we could develop a reporter system for improved protein delivery to cells. We have seen different types of proteins, GB1, PDC domain, ubiquitin. I didn't show. I kept that slide away today. And so on. We see that there is a multiplicity of these protein resonances and so on. And we could see the specific interaction demonstrated in the case of PDC domain in terms of the ligand protein binding. And we also were able to determine the protein structure in cells. That was kind of hardly shown by the research community, NMR-based research community before. And the protein IDP, the intrinsically disordered protein, prothymosin alpha, shows a lot of features in mammalian cells, and that allows the state-of-art, the various NMR experiments that can be performed, and we can propose various molecular mechanism involved in their cancer progression and so on. I also would like to, because I have seen when I looked at the schedule today, there are a lot of computational works that you are doing in your department and so on. It's so amazing to see. But in terms of the research, just because I saw a kind of main protease, someone is presenting a presentation with in terms of related to COVID research, for example. We also did a lot of MPRO, main protease related research experimentally. It's one of the projects I was involved in was for the spike protein, and not the whole spike protein is too big for NMR. So what we do is we take the RBD, receptor binding domain, of this one, right? And its interaction with the receptor human ACE2, and now what we look at is like the binding interface. So the idea of the project back in 2020, soon after COVID was started, what we did is we collected, pooling a lot of FDA approved drugs already available commercially available and so on. We tried to test everything. In fact, to look at, first we wanted to see whether any of these small molecules interacts with either of these proteins. We also titrated them to the Nmr. So I was not directly part of this main protease research. So I was mainly looking at this. That's why I would like to show this one. No? So now the idea is like, any of the FDA approved drugs before all the vaccine developments. So we wanted to see any of the molecules interacts. If it interacts, whether it can prevent the association of these, so RBD as well as ACE2 receptors. So if you can do that, probably you can delay or prevent getting COVID and so on. That is the idea, right? And that's still a lot of pharmaceutical industries like Lonza, Novartis, Pfizer, a lot of people, Johnson & Johnson, everyone are actively involved in also the small molecule research, anti-COVID research too, right? So just want to say, okay, these are many things what we can do with Biomolecular Nmr. And now a lot of things one can do in personally. These are some of the projects I am interested. What I'm trying to show here is like, there are, which I already tried back in Zurich, and also some in collaboration with some of the labs in India also. We can do a lot of detailed investigation. We have a lot of Ayurvedic medicines. Since in organic chemistry, in chemistry, that there are a lot of natural compounds also, right? So which are, you can extract a lot from also Ayurvedic medicines and so on. Sometimes you don't know the mechanism of the certain drugs, certain medicines, Ayurvedic medicines. The modern medicine we know because it came through clinical trials and so on. People more or less know the understanding, how these molecules are interacting, but many things are unknown in terms of our traditional treatment methods, right? So we can do that. So I already have a lot of ideas and initial results, so I wanted to explore it. But I just want to tell you that we can do a lot of interesting, cool things, for example, right? And the COVID-19 research, as I've shown in the previous slide, people can do insulin MR. So another important project I was involved in is like, structure of the KRAS protein. This is one of the important cancer-related proteins. This KRAS, I'm still doing it, part of this research here. I actively was involved back in Zurich also. So this particular protein is involved in more than 30% of the cancer cases, right? More than 30% of the cancer cases, this KRAS protein is mutated. And especially in pancreatic cancer, this particular protein is mutated in almost 90% of the cases. So this is a very important protein. So far, there is no drug, anti-cancer drug against KRAS. This is such an important protein, very notorious. Many pharmaceutical industries are trying to develop anti-cancer drug by focusing on protein. So far, there are no direct drugs. Some of the molecules are in clinical trials and so on. And which actually started in collaboration with Novartis. Novartis is one of the world famous pharmaceutical industries, and there are a lot of collaborations and so on. But we are I'm highlighting this for the students, just to tell that you see there are a lot of opportunities in any area, computational approach, experimental approach, and so on. A lot of things we can do in terms of structural biology. So for that, we should have a very strong chemistry background. If you have very strong chemistry background, you can do a lot of biochemistry, chemistry, pharmaceutical chemistry, and so on. Okay. With this, I would like to acknowledge a lot of people involved in the project back in Göttingen, Germany, back in Zurich, and so on, right? And and thanks a lot for your kind attention. And this was a group I was working on, just the picture just before I left Zurich in August. And we were mainly located, I think I shown in the very first slide, we were located somewhere here in the Hongerberg campus. It's such a beautiful campus. I hope at least, I wish some of you can do some of your research, part of your research at least. If you do PhD in India, go for postdocs abroad, try to get positions in important labs and, and so on. If you let me know, I can also do, whatever I can to support personally. There's no problem with that. I'm happy to help. And thanks a lot again for your kind attention. I understand I took a bit longer than expected. Yeah. Thanks a lot. And with this, I also would like to take this opportunity to wish you all Merry Christmas and Happy New Year coming. And I'm happy to take questions. Any questions if you have time, please go on.
Thank you, sir. It was indeed a great beginning for a session. Now our session is open for discussion.
Hello, sir. Am I audible?
Yes, please. Yeah.
Yes. Sir, my name is Arun Thomas, faculty in the Department of Chemistry, St. Stephen's College, Uzhavoor. I have some doubt in the instrumentation part. So, while measuring the NMR, the solubility is an important thing. So, in this biomedical NMR also, is the solubility has any problem? Like, or you are measuring, we have the possibility of solid state NMR, something like that?
It depends, right? I mean, what kind of molecule you are measuring? So usually, most of the when you do bio, so I have to tell you this, no. So when we do biomolecular NMR, in my case, for example, so proteins. They don't. I mean, they like organic solvents, but we need to mimic the physiological state, in our blood in cells. So this is an aqueous environment, right? So we provide the buffer conditions like a suitable NMR buffer, normally around pH 7 to 7.4. Some people do, depending on the condition, pH 6.5, 6.8, and so on. Something, let's say, on average, we could take from pH 6 to pH 8, somewhere in between, which is most of the time we try to go for pH 7.4 just to mimic the physiological conditions. So the proteins are soluble in that. Now, when you bring some small molecules, certain molecules are soluble in water, but some proteins are not. Some molecules are not. Certain molecules like aromatic, especially those with aromatic rings and hydrophobic like CS3 groups or even aromatic phenyl ring and so on. So they are hardly. I mean, sometimes they are sparingly soluble in water. So what we do is we provide a little bit of DMSO. So the molecule will be dissolved in DMSO, and then DMSO and water, they will mix, right? So somehow you enhance the solubility. There are a lot of situations that we were really limited by. That was your question, right? Sorry,
sir, I didn't get you.
So
I asked which solvent system you are preferring for biomedical NMR.
Yeah, that was my point. So I explained a bit too much. So we are limited to use aqueous solvents like the buffer, basically water, right? So that's because protein, we are working with proteins, biomolecules, whether it is RNA, some people work with RNA a lot, but I mainly work with proteins and peptides with small molecules, of course. So these proteins and peptides, just to mimic the physiological conditions, we provide pH 7.4 buffer. So these buffers are, of course, in water, right, in an aqueous condition. But when you do the protein ligand interaction studies, or when you want to determine the structure of a complex and so on, or when you do the NMR titration, you add the small molecules. Some of the small molecules are soluble in water, depending on their functional groups like phenyl ring or CS3 groups and so on. Sometimes they are not 100% soluble in water. In that case, we use a little bit of DMSO to make the stop concentration. So you dissolve everything in DMSO, then you add this little bit, a small fraction of this into the protein in water. So you have a kind of mix of water and DMSO. So because of this DMSO water interaction, there will be a little bit of enhanced solubility. However, if you want to bring this small molecules in a large concentration, it might precipitate in water environment. That's only a limitation we have. Because we cannot do 100% of DMSO and protein, because all the proteins will precipitate at the end of the day.
Okay, sir. Thank you. I have one more question. So usually for small organic molecules, we need to run the sample for 8 to 32 number of scans.
Yes.
Number of scans.
You're talking about proton NMR, right?
Yeah, proton NMR. And how much time will take for measure and biomolecular NMR? I mean, number of scans.
Yeah. So that again depends on. Okay, let me ask you a question. So, what's the concentration of your small molecule? What's the concentration of your sample?
Sorry, sir.
What's the concentration of your sample?
I mean, I just want to know how much time it will take. So if you have enough concentration for protein proton NMR, it will take you some 5 to 10 minutes. I just want to know how much time it will take for a biomolecules, because the
So,
molecule is large.
just put it like
Because
this,
the-
you know, so we use 100 micromolar sample, let's say, a normal protein. That means depending on the type of the number of amino acids, you know, sometimes it's a 5 milligram, you know, 5 more millimolar solution, sometimes it's 1 millimolar solution, sometimes it's micromolar concentration. So depending on the number of moles, of course, but usually we, most of the proteins, in general, I can talk for a 1D spectrum, we need like maybe 2 minutes for a 1D spectrum or 1 minute, a proper 1D, but 1D, of course, we cannot analyze, you know, it's so complicated, so overlap. So we always need a 2D. The normal 3D, a 2D experiment takes like 30 minutes to 1 hour. 30 minutes to 1 hour a 2D experiment. With that, you can do a lot of experiments. And if you want to do, so a 1D takes 1 minute, a 2D takes 30 minutes to 1 hour. And if it's a 3D experiment, it takes 1 day minimum. Okay. At least 1 day. And depending on the concentration of the protein, it changes. You know, I'm talking now on average for normal proteins.
Okay, sir. Okay, sir. Thank you.
Okay.
And one of our students has raised one question. I shall read. What is the possibility
please,
of
course, just go. Could you
what
repeat
is
again?
she has raised the question. What is the possibility of solid state NMR for the analysis of biomolecules? Is there any possibility? Solid state
Yes.
NMR and
there are a lot of research. Even I know, during my time in NCL Pune, that was back in 2007, right? And Bindu also was there for her master thesis, you know, when I was working as a participant there. So we were doing solid state NMR of inorganic materials. So my point is like, you know, my interest is also in solid state NMR, now especially at this stage in biomolecules. So I also do a biomolecular solid state NMR here. What are the possibilities, especially, for anything that is soluble? You cannot use solid state NMR. You know, it should be a solid part material. Sometimes there are certain proteins that forms a fibrils. In Alzheimer's disease, Parkinson disease, this kind of, misfolding diseases, these proteins deposits. In one of the introduction slides I mentioned, right, these proteins deposits, but this cannot be solubilized in water at all or in buffer at all. So in those conditions, if you want to study the structural states, just if you want to understand what's the molecular mechanism involved in their kind of precipitation or fibril formation, it's called fibrils, protein fibrils. So any Alzheimer or Parkinson brain, patient's brain contains a lot of these kind of precipitates, like a protein precipitates or protein deposits. You can study this by means of solid state NMR. One of the methods I'm saying for biomolecules. Okay, and another thing is I like to study membrane proteins. Membrane proteins means, in our cell membranes are composed a lot of lipids, and when a protein, membrane attached protein, membrane proteins are very important for a lot of transport from extracellular environment to the intracellular environment. For example, the calcium transporter, sodium potassium pump, for example, everything works through membrane protein systems. So if you want to study the membrane protein, solution NMR is very limited. You can use solution NMR to some extent, but mostly this can be studied using solid state NMR. So to study membrane proteins, to study protein deposits or protein fibrils, there are a lot of applications for solids.
And there is one more question from the audience part. Sir, more about the application of vinblastine into molecules inhibition.
Vinblastines what exactly is the question? So vinblastine tau interaction?
Molecules inhibition. Vinblastine into molecules inhibition.
Okay, let's go there. I didn't get exactly what it is. Is it somewhere here? Can they ask the question? Is it possible for them to ask? I didn't get exactly what.
Yes, sir.
Yes.
Sir, can you say more about the applications of cancer drug vinblastine in inhibiting tau molecules that cause Alzheimer's disease?
Okay. So vinblastine doesn't have any direct application in Alzheimer research. Okay. So I introduced vinblastine here. Of course, this project is mainly Alzheimer research project. But main idea was, so tau binds at somewhere. We didn't know where it binds. But when you look at this, but the molecule is the same. Microtubule is the same. All these molecules bind at the microtubules, and they have binding sites which is already known. So I just wanted to know whether, yeah.
Hello, sir.
Yeah. Tell me. Yeah.
Sir, sir, it was not. Sorry, can you repeat it again a little bit?
So the main idea of this part was, the binding site of tau is not clear. And now there are some small molecules like vinblastine, taxol, and so on, for which the binding sites are already known. However, these are anti-cancer drugs mostly, okay? So these are not anti-Alzheimer drugs. So there is no direct correlation of vinblastine or taxol with Alzheimer disease. However, tau, I was thinking like, tau might share the binding sites with one of these molecules. It's only about the binding site. Apparently, by doing all these competition experiments as well as HTB-NMR experiments, I found out that also the results from the mass chemical cross-linking as well as mass spectrometry, we realized that, okay, tau is binding at the interdimer interface of alpha-beta tubulin heterodimers, where vinblastine is binding. But now when you bring vinblastine to an Alzheimer as an anti-Alzheimer drug, it won't work. I think that was a question, right?
Yes, sir. It's clear. Thank you, sir.
You're welcome.
I think we can wind up the session now. So, good morning all. I'm Dr. Bindu. It is my immense pleasure to propose vote of thanks to Dr. Harindranath. Actually, myself and Dr. Hari were together in NCL Pune. He was talking about it. When we thought of organizing this conference, actually the persons who came into my mind were those in NCL, of course. So when I approached Hari, he readily agreed to our invitation. And though we were in not contact for long years, but he readily agreed to our invitation. And it was late night in December, though it was only his concern, I guess. But still, he agreed for our invitation. And on behalf of St. Stephen's College, Uzhavoor, and on behalf of the staff and students of the PG Department of Chemistry, I extend a heartfelt thanks to Dr. Harindranath Kadavath for this wonderful talk. Thank you, Dr. Hari. Though the sessions are yet to continue, I would like to take this opportunity to extend my heartfelt thanks to Professor A.B. for giving me full freedom to arrange this conference. And also I extend my thanks to all the faculty members in the PG Department of Chemistry. Also, I would like to thank Dr. Priya Mary Abraham, who has accepted to judge the sessions, presentation competitions, and the student coordinators, Ansi, Ria, and Alinta of First MSc Chemistry. Also, I would like to thank and apologize all the participants who are taking part in this conference. Though the time has extended half an hour, I guess. So they are really patient and they are with us. So I thank all the participants who took effort in registering to this event and who are going to present in this conference. Thank you all and have a good day. Thank you.
Thank you, Bindu ma'am. Now we start our paper presentation. First, I would like to invite Jogi Alexa to present his work. Sir, please.
Hello. Hello.
Hi, sir.
Can you hear me?
Yes, sir.
I would like